So our first grant recipient is Dr. Ronald Cohn, President and CEO of the Hospital for Sick Children, SickKids in Toronto, for his project entitled Simultaneous DMD Gene Editing and Upregulation to Restore Protective Levels of Full-Length Dystrophin in the Treatment of DMD Duplication Mutations. Dr. Cohn, I'll pass it over to you.
Thank you so much, Lisa. Let me pull up my presentation. All right, good evening, everyone. And first of all, let me echo some of the words that you have heard from literally everyone so far in thanking you for your support, particularly during this, what clearly has been one of probably the most difficult years we collectively and individually had to go through. And I say this on behalf not just of my laboratory and my lab members, but really collectively on behalf of so many scientists. The fact that you continue to rally to support our science, our research, is something that I am in awe of and have been in awe of throughout the entire year, through so many people coming together to support not just Duchenne muscular dystrophy research, but so many other areas of foundations and funding. Because I've said it many times before, it's parents and individuals like you who allow us, who are scientists, to go forward with some of our creative thoughts that are not necessarily ripe enough to be funded through CHR grants or the more traditional grants. And I think foundations like Jesse's Journey have become the absolute anchor of creative science in our area. So wholeheartedly, thank you everyone for supporting not just my laboratory, but really everyone you will be hearing from today. So I'm not going to spend much time on what Duchenne muscular dystrophy is, because you know this as at least as good as I do. But what I do want to spend some time on is the project that you have been helping us to get off the ground now. So there are about 11% of patients who have Duchenne muscular dystrophy who have a duplication of the dystrophin gene. And these duplications somewhat present a unique therapeutic challenge if you think about genetic and gene therapy and genome editing therapies. And as many of you know, my laboratory has been working now for almost 7 years already on genome editing and the CRISPR technology. And what the CRISPR technology really provides you with is an opportunity to perform genetic or genomic surgery, meaning you have the ability to cut out faulty parts of a certain gene and thereby try to find mechanisms of how you can repair those. Now, if you think about a duplication and you think about the ability to go into a genome and cut, then you would think, wow, that actually is probably one of the best ways of using the CRISPR technology, because you just go in there with 2 scissors and you cut out the duplicated material, and then you end up with what we would call the normal wild-type gene of dystrophin. And while that's the case, my laboratory has several years ago invented a methodology where instead of using 2 scissors or 2 guides, we're using 1 single guide that is cutting in part of the normal gene as well as in part of the duplicated gene, which has the same sequence, and thereby gives you then a wild-type gene. And the advantage of this, and the reason why this was really a phenomenal job by one of my previous graduate students and my research associate, is that if you think about how to deliver 1 scissor or 1 guide to initially a mouse and then eventually a human, it is much easier to do that, has probably significantly less side effects, and that's why this is a very elegant method. And we use this method in a mouse model that we created with a duplication of exon 18 to 30, where we injected this 1 guide into the vein of a mouse, where it would then lead to systemic distribution, in order to see whether we can restore the wild-type normal full-length dystrophin in these mice. And that's exactly what we were able to see. You see on the very right side, the untreated duplication mice that have no dystrophin expression, like you would see in a boy with Duchenne muscular dystrophy. And in the middle of the panel, you can see that in one of the leg muscles, as well as in the heart, the application of the one single guide through an adeno-associated virus gives you quite robust expression of dystrophin protein, which again is a wild-type full-length dystrophin. That's different from some of the gene therapies that are currently in clinical trials where we are giving only a smaller version of dystrophin. So here we are restoring the full length of wild-type dystrophin. And although, as you can see, I'm going to go back for one more second, while you see fairly robust expression, you can see it's not all of the muscles, neither in the heart nor in the skeletal muscles of the extremities. But you can see that despite this, we see significant improvement in the strength of the mice, in the scar tissue fibrosis of the mice, and we also see significant improvement in the overall expression levels of dystrophin that then correlate to some of these strength and scar metrics that I'm showing you here. Now, what you are now helping us to figure out is the following. While we see fairly good results with our initial experiment, the idea we are having right now is, can we not only cut out the duplication, but can we use the CRISPR technology in another aspect by trying to enhance the expression of the protein levels? So what you can do with the CRISPR technology is you can inactivate the scissors and just try to make sure that the guide is binding to the promoter that enhances the gene expression and the protein expression. And we are trying to think about this dual approach by cutting out the duplication on one hand, and at the same time pushing the expression of dystrophin so that our levels of dystrophin are even better than what I've shown you before. And that's exactly what you can see here. It's a complicated slide. So the idea is really that, as I said, we anticipate that we can increase our dystrophin levels by an additional maybe 30 or even 40 to 45%. And this just shows you that we can actually successfully do this. And on the very right side, you can see there is a combination of a guide 9, 10, 11, and a fairly robust dark band of dystrophin expression that is proof that in the heart cells of mice, we can demonstrate that the expression of dystrophin can actually be upregulated through this methodology. So what we are now going to do is we are trying to use these guides that I showed you, 9, 10, 11, and we are going to incorporate them together with our scissor guide to make sure that we upregulate dystrophin on one hand, cut the dystrophin duplication out. And what we are now going to do is we will start, and we actually have just started this 2 weeks ago. I wish I would have results, but we need to usually wait 4 weeks until we can look at the mice. So we are just starting with a proof of concept experiment where we just inject the muscle. And if successful, similar to what I showed you in the cells earlier, then we are going to do the same experiment and inject it into the vein and make sure that we can evaluate this in all of the body of these mice. So that's really the gist behind the research project that we are doing. I do want you to know, while we are focusing initially on this duplication mouse, the idea of what we are trying to do here is really something that would be able to be used in all other genome editing applications, particularly for deletions, as well as for other additional targets. So what you're supporting here, while we are just focusing on a small area of research, has huge implications if it works, and hopefully it will work. If it works, not just within the DMD field, but is a technology that can even be used for other rare diseases where improving the gene expression and protein expression of a diseased gene can be of benefit. So I'm going to stop here, and I'm happy to answer questions later, but I do want to at least point out a couple of the people who are doing the important work. So you can see here, I hope you can see my mouse. This is Elle, who is one of my graduate students who has done all the work until the moment when we submitted the research grant to you. And I'm trying to find my student. There he is, just above my head here. This is Matthew Rock, who is doing all these experiments I just showed you. So thank you again very much for your support. We couldn't do it without you. And I'm excited and looking forward to tell you more, hopefully next year, about some of the successes of what we are doing.